mitotic divisions that takes place during the development of male gametophyte in the pollen chamber and archegonial chamber respectively for the formation of 80 male gametes. For the formation of 80 male gametes in cycas, definitely before pollination 2 mitotic divisions, after pollination 2 mitotic divisions occur. So, in total 4 mitotic divisions occur, but before that 2 mitotic divisions, after that 2 mitotic divisions. So, definitely answer is 40 and 40, fourth option. In a cycas ovule, nucellus contributes for the formation of following structures. Nucellus contributes for the formation of following structures. Nucellus contributes for the formation of nucellar beak, correct. Nucellus contributes for the formation of pollen chamber, correct. Nucellus contributes for the formation of archegonial chamber, correct. Nucellus contributes for the formation of pollination drop is also correct. And nucellus contributes for the formation of perisperm is also correct. So, fourth option must be 1, 2, 3, 4, 5. So, in cycas ovule, nucellus contributes for the formation of following structures before fertilization. Before fertilization it is so perisperm will not come. Perisperm comes only after fertilization. So, before fertilization means nucellar beak comes from nucellus, pollen chamber comes from nucellus, archegonial chamber comes from nucellus, pollination drop comes from nucellus, but perisperm also comes from nucellus, not before fertilization but after fertilization. So, in cycas ovule, nucellus contributes for the formation of following structures before fertilization. Answer is nucellar beak, pollen chamber, archegonial chamber, pollination drop, but perisperm also remnant of nucellus, but the question here is before fertilization. So, fifth option will not come. 1, 2, 3, 4. So, fourth option is the answer. Generations represented by mature seed, cycas seed. Every seed of cycas shows 3 generation parts. 1. Parental sporophyte. Parental sporophyte structures are seed coat and perisperm, remnant of nucellus. Gametophyte. The gametophytic structure is female gametophyte or endosperm. Female gametophyte or endosperm. Third, new sporophyte. New sporophyte structure is embryo. So, every seed of cycas shows parts of 3 generations. Parental sporophytic generation are seed coat and perisperm. Gametophytic generation structure is female gametophyte or endosperm. New sporophytic generation structure is embryo. So, the question is generations represented by mature cycas seed are old sporophyte, gametophyte and young sporophyte. Old sporophyte are seed coat and perisperm. Gametophyte is female gametophyte or endosperm and young sporophyte is embryo. Old gametophyte sporophyte is wrong. Old and young sporophyte is wrong. Old and new gametophytes is wrong. Old sporophyte, gametophyte and young sporophyte. First option. Arrange the following in a correct ascending order with reference to cycas. Minimum number of microspori on a microsporophyll. Minimum number of microspori will be 1 to 6 enter. So, 1 to 6 will be so. Next, number of mitotic divisions occur in the development of male gametophyte 4. Minimum number of meiotic divisions occur in each megasporophyll 1. Maximum number of egg cells produced by a female gametophyte. Maximum number of egg cells female gametophyte will be 2 to 8. So, answer is 3, 2, 4. 3, 2, 4, 1. 3 is first. Minimum number of, it is ordered in ascending order. Minimum number of meiotic divisions occur in each megasporophyll is 1. Next, 2. 2 is number of mitotic divisions occurs in the male gametophyte 4. Then 4. Maximum number of egg cells produced by a female gametophyte is 8 and 1. Minimum number of microspori in each will be around 70 to 100. So, first option. Identify the ascending order of the following structures in cycas based on their number. Minimum number of ovules per each megasporophylls, minimum is 2. minimum is 2. This is also you have to arrange in ascending order. So minimum is 2, first one. Number of meiospores formed in an ovule, number of microsporangia per sorus, number of layers in the integument of mature ovule. So answer is 1, 4, 2, 3. 1 is minimum number of ovules per megasporophyll. Number of layers in the integument of mature is 2. Then single integument, so 2, 1. Then fourth option, number of layers in the integument of mature ovule. Then, so first option, fourth option, second option, third option. So 1, 4, 2, 3 is the correct ascending order of the following structures of cycas based on their number. The ratio of mitotic divisions during the development of male gametophyte before and after pollination in cycas. In cycas, microspore undergoes 2 mitotic divisions before pollination. Before pollination. 2 mitotic divisions after pollination. So 2 is to 2, that is 1 is to 1. So the ratio of mitotic divisions that occur before and the ratio of mitotic divisions during development of male gametophyte before and after pollination is: before pollination, microspore divides and produces an antheridial cell and a prothalial cell. Antheridial cell divides and produces tube cell and generative cell. Till that it occurs before pollination. After that, generative cell divides and produces stalk cell and body cell. Then body cell divides and produces male gametes. So answer is 1 is to 1. 2 is to 2, that is 1 is to 1. Haploid endosperm is absent in gymnosperms. Assertion: haploid endosperm is absent in gymnosperms. Female gametophyte itself acts as endosperm in gymnosperm. Haploid endosperm is absent in gymnosperm is wrong. Why? Because in gymnosperms, endosperm or female gametophyte is haploid, which gives food for developing microspores or whatever it may be, developing archegonia or many other things. Female gametophyte itself acts as endosperm in gymnosperms is correct. So A is false, R is true. A is false, R is true. Fourth option. Largest structures in the plant kingdom are produced in cycas. 3 largest structures are produced in the plant kingdom in cycas. One is largest ovule. The other is largest egg cell. The third is largest male gamete. So 3 structures in cycas are produced in largest amounts: largest ovule, largest egg cell, largest male gamete. So here the question is given: what are the largest structures formed in cycas? Last cell of male gametophyte, that is male gamete, correct option. First cell of male gametophyte, wrong. Last cell of female gametophyte, correct, egg cell. And first cell of female gametophyte, wrong. So answer is 1 and 3, second option. From the following, identify the simultaneously formed cells during the formation of male gametophyte of cycas. Simultaneously means they are formed one after the other. Body cell and vegetative cell are formed, not togetherly formed. Antheridial cell and vegetative cell not togetherly formed. Generative cell, stalk cell not togetherly formed. When a body cell divides, both the male gametes are formed simultaneously. So from the following, identify the simultaneously formed cells during the formation of male gametophyte of cycas is: 2 male gametes are formed simultaneously. First option. The total number of types of cells formed in the male gametophyte of cycas when it is developed in the pollen chamber. The total number of types of cells formed in the male gametophyte of cycas when it is developed in the pollen chamber. When it is developing a pollen chamber, it goes to the pollen chamber in 3-celled stage: prothalial cell, generative cell, tube cell. So generative cell divides and produces stalk cell, body cell. Body cell divides to produce 2 large multiciliated top shaped motile male gamete. So question is: the total number of types of cells formed in pollen chamber is stalk cell, body cell, male gametes. So, definitely 3. First option. Largest in the plant kingdom produced by cycas are sperms, male gametes. Microspores or pollen grains are not largest. Egg cell is the largest structure found in cycas. So, first and 3 are the absent. Second option. Cycas revoluta is popularly called Sagocycas. The speciality of this cycas is it is a Japanese species introduced in India. As an ornamental plant. The special feature is in Cycas revoluta, xylem vessels are present. When you take the TS of leaflet, in Cycas cercinalis, palisade tissue is restricted only to the mesophyll region. It does not extend to the midrib region. But in Cycas revoluta, palisade tissue is even extended into the midrib region. Palisade tissue is present even in the midrib region. In cercinalis, it is restricted only to the mesophyll region. It does not extend into the midrib region. But whereas in revoluta, it is extended even into the midrib region. Natural occurrence in India is wrong. It is a Japanese species introduced in India as an ornamental plant. So, natural occurrence is wrong. Cycas revoluta's characters are xylem shows xylem vessels. It is the only species in Cycas where xylem vessels are present. Palisade tissue is extended even into the midrib of leaflet is correct. In Cycas cercinalis, palisade tissue is restricted only up to the mesophyll region. Natural occurrence in India is wrong because it is a Japanese species introduced in India as an ornamental plant. So, answer is 1 and 2. First option is 1 and 2. Leaf modifications in Cycas are. In Cycas the leaves are modified. Leaves in Cycas are said to be dimorphic. Leaves. So, leaves, scale leaves and foliage leaves. Scale leaves are triangle in shape surrounded by brown hair-like structures called ramenta. Presence of ramenta is a fern character in Cycas. Foliage leaf shows a rhomboidal leaf base and a hard central rachis. On both sides of the rachis, you will find numerous leaflets. The lowermost leaflets are modified into spines. So, definitely in Cycas, the leaf modifications are one is scale leaves. The other is spines. Scale leaves are surrounded by brown hair-like ramenta is a fern character in Cycas, and other is scale leaves. One is spines. Why? The foliage leaves, some of the foliage leaves present at the base are modified into spines. So, leaf modifications in Cycas are spines and scale leaves. Phyllodes are not found in Cycas. So, the question leaf modifications in Cycas are spines and scale leaves. Fourth option. Cycas has dimorphic roots, dimorphic leaves, dimorphic leaflets, dimorphic roots. Definitely dimorphic roots because roots in Cycas are two types, adventitious roots and corylloid roots. Leaves in Cycas are also dimorphic, scale leaves, foliage leaves. Then dimorphic leaflets. Leaflets are also dimorphic. Why? Some leaflets are complete and some leaflets are modified into spines. So, Cycas shows dimorphic roots, adventitious roots and corylloid roots. Cycas shows dimorphic leaves, scale leaves and foliage leaves. Cycas shows dimorphic leaflets. Some leaflets are well organized leather shape with only midrib without lateral veins present on the stem, and some leaflets are modified into spines. Lowermost leaflets are modified into spines. So, Cycas has dimorphic roots, dimorphic leaves and dimorphic leaflets. 1, 2, 3. Answer is 4. These cells of Cycas male gametophyte are able to divide. Body cell divides to produce male gametes. Antheridial cell divides to produce generative cell and tube cell. So, whenever microspore divides in Cycas, first it produces prothalial cell and antheridial cell. Prothalial cell is inactive, antheridial cell is active. So, this cell does not divide. Antheridial cell divides to produce tube cell, generative cell. Tube cell forms pollen tube. Generative cell again divides to produce stalk cell and body cell. Stalk cell is not dividing. Body cell divides to produce two male gametes. So, what are the cells are dividing, what are the cells are not dividing, you can see in the tabular form. Microspore pollen grain producing different types of cells by 4 mitotic divisions. So, here the cells of Cycas male gametophyte which can able to divide are body cell, antheridial cell. Male gamete is formed by the divisions of body cells, so it does not show any type of division. So, answer is 1 and 2. Transfusion tissue is present in Cycas. In Cycas, midrib is present, lateral veins are absent. Cycas leaflet resembles like this. Every vascular bundle surrounded by special parenchyma tissue. In this parenchyma tissue, you will find very closely arranged xylem trachea-like cells called primary transfusion tissue. From this primary transfusion tissue, another group of very compactly arranged cells are formed. From primary transfusion tissue, special group of very compactly radially arranged cells are formed running from vascular bundles to margin of leaflet, which is called secondary transfusion tissue. Primary transfusion tissue and secondary transfusion tissue ultimately forms transfusion tissue. In Cycas leaflet, lateral veins are absent. So, the lateral conduction is done by transfusion tissue. So, transfusion tissue compensates the absence of lateral veins. So, transfusion tissue, how it is present? In the parenchyma cells, it is present as primary transfusion tissue, compact arranged xylem trachea-like cells with bordered pits called primary transfusion tissue. In the primary transfusion tissue is connected to radially elongated special xylem trachea-like cells with bordered pits called secondary transfusion tissue. Primary transfusion tissue and secondary transfusion tissue ultimately forms transfusion tissue that helps in lateral conduction and compensates the absence of lateral veins. So, transfusion tissue is present in which part of Cycas means Rachis does not show. Rachis is identified by, Rachis identification is, it shows numerous vascular bundles in an inverted omega pattern. So, numerous vascular bundles arranged in an inverted omega pattern is the identification feature of Cycas rachis. Coming to the root. Root is normal root with many layered pericycle and coralloid root with 1 or 2 layers pericycle. Normal root shows tetrarch and exarch condition like dicot root, but coralloid root shows triarch and exarch condition. Those are the identification feature. Coming to the leaflet, leaflet only contains transfusion tissue, that is helping in lateral conduction due to the absence of lateral veins or compensating the absence of lateral veins. So, answer is only 3, fourth option. Open vascular bundles are seen in. Open vascular bundle means every vascular bundle in Cycas stem and root are like this. They show xylem on the lower side and phloem on the upper side with cambium in between xylem and phloem. Whenever cambium is present in between xylem and phloem, it is called open vascular bundle. So, in Cycas, you will find open vascular bundles not in Cycas root, because Cycas root is radial vascular bundles, open vascular bundles are not there. It is found in Cycas stem. Cycas stem shows open vascular bundles with cambium in between xylem and phloem. Another important feature is Cycas leaflet. In Cycas leaflet, the vascular bundle is conjoint collateral, open, with pseudomesar condition. Conjoint means xylem and phloem jointed to form one bundle. Collateral means xylem and phloem are present in the same radii, one above the other. Open means cambium is present in between xylem and phloem. If you observe the cycas leaflet vascular bundle, it shows a centripetal xylem like this. It shows two centrifugal xylem like this, and below this xylem you will find phloem. In between xylem and phloem there is a non-functional cambium. So leaf containing open vascular bundle is a special feature. Normally leaves show closed vascular bundles. So leaf showing open vascular bundle is a special feature. So open vascular bundles are seen in main cycas stem and cycas leaflet. Cycas leaflet. So 2 and 3 is the answer. That is second option. Cycas male plant shows. Cycas male plant speciality is it produces male cone. Male cone is formed by the modification of apical bud. Whenever male cone is formed from apical bud, that means apical bud converts into male cone. Definitely the growth of the main stem stops, and that is called sympodial growth. That is called sympodial growth. So cycas male plant always shows sympodial growth. Whereas in female plant, the apical bud is not converted into female cone. Only megasporophylls are loosely arranged. It is called monopodial growth because main axis grows indefinitely. So cycas male plant always shows sympodial growth. In male plant, male cone is present, which is formed by the aggregation of microsporophylls. Every microsporophyll shows a pointed spinous tip called apophysis. Many sori on the ventral side and many other structures. So that is about cycas male plant always shows sympodial growth. Why sympodial growth? The main axis stops its growth. Why main axis stops its growth? The apical bud transforms into a special structure called male cone. So only 2 is correct, sympodial growth. Structure of cycas, feature and feature 1 and feature 2. Seed, perispermic, polyembryonic, correct. Embryos are more, perisperm will be there. Female gametophyte has archegonia, 2 neck canal cells. Now archegonia neck canal cells are completely absent. Only 2 neck cells are present. Male gametophyte has ciliated gametes and prothelial cell, correct. Microsporangium born individually use sporangiate, wrong. So answer is 1 and 3. Seed, perispermic, polyembryonic, correct. Male gametophyte has ciliated gametes and has prothelial cell is correct. So 1 and 3 are correct. Cycas structure, size and character. Megasporophyll 25 centimeters leg has ramenta is correct. Microsporophyll 3.5 centimeters had individual hair is correct. Male cone has few microsporophyll is wrong. Foliage leaf has tendrils is wrong. So 1 and 2 is correct. So first option is always correct. Arrange the following structures in cycas ovule with its female sex organs in centrifugal manner. Centrifugal manner means from center to periphery. Center to periphery means first haploid tissue female gametophyte. First haploid tissue female gametophyte. Third option. Sorry, first option, third. Next comes 4, archegonial chamber. Just above the female gametophyte or endosperm, archegonial chamber. Above the archegonial chamber, you will find nucellus. And above the nucellus, you will find sarcotesta. So answer is 3, 4, 2, 1. First option. Arrange the following formation of the following dividing cells of male gametophyte in correct sequence of their formation. How they are formed. First formed is microspore. Always microspore is first formed. Then formed is antheridial cell. Microspore divides to form antheridial cell. Then antheridial cell divides to produce generative cell. Generative cell divides to produce body cell. So first is microspore 4. From microspore antheridial cell is formed 3. Then generative cell 2. Finally body cell 1. So answer is 3, 4, 3, 2, 1. Arrange the following structures of a species of cycas in a sequence of increase in their number. Megasporophylls, then comes microsporophylls, then comes foliage leaves, and finally scale leaves. Increase in their number, that means less to more. Megasporophylls less in number, then microsporophyll number is more. So 1 first megasporophyll, then microsporophyll next, then foliage leaves, and maximum number will be scale leaves. So minimum number will be megasporophylls. Next to megasporophylls, next is microsporophylls. Next microsporophylls, foliage leaves. Foliage leaves means performing photosynthesis. Large number is scale leaves. So arrange the following structures of a species of cycas in sequence of their increasing number. Increasing number means less to more. Megasporophylls, microsporophylls, foliage leaves, scale leaves. So 1, 3, 4, 2 is the answer. Arrange the following in cycas with gradual increase in their size. Gradual increase in their size. Ovule, microsporophyll, megasporophyll, male cone. First microsporophyll will be less, then ovule, then megasporophyll, then male cone. So first is 2 microsporophyll, a little bit more size is ovule, and a little bit more size than ovule is megasporophyll, and above all the large is male cone. So 2, 1, 3, 4. 2 is microsporophyll, 1 is ovule. The largest ovules are even present on megasporophyll. So definitely structure of megasporophyll is more than ovule, male cone. So microsporophyll size is a little bit less. Next is ovule. Next is megasporophyll. Final is male cone. So answer is 2 first microsporophyll, 1 next ovule, megasporophyll 3 and 4 male cone. Second option. Arrange the formation of the following sequence during the development of male gametophyte. First formed is generative cell. First formed is generative cell. Just now I wrote microspore divides and gives rise to antheridial cell and prothalial cell. Antheridial cell divides and gives rise to tube cell, generative cell. Generative cell divides and gives rise to stalk cell, body cell. Body cell divides and gives rise to male gametes. So sequence is first microspore, then antheridial prothalial cells, then tube cell, then generative cell, then stalk cell, then body cell, and then male gamete. So according to this question, the sequence is first is always prothalial cell. So third. Next is generative cell. Prothalial cell and antheridial cell are formed. Antheridial cell divides and gives rise to generative cell. So second one comes. Then generative cell divides and produces stalk cell and body cell. So body cell will come now. And finally body cell divides and gives rise to male gametes. So answer is 3, 2, 1, 4. First is prothalial cell, then is generative cell, then is body cell, finally male gametes. 3, 2, 1, 4 is the correct sequence of formation of various spores during the development of male gametophyte in cycas. Arrange the following structures of ovule with the development of female gametophyte present in centripetal manner. Centripetal means periphery to center. Periphery to center. First sarcotesta, then nucellus, then inner sarcotesta, then endosperm. So fourth option 1, 4, 2, 3. 1, 4, 2, 3. First sclerotesta, then inner sarcotesta. So every ovule of cycas resembles like this. It shows a thick sclerotesta, 2 sarcotesta, pollen chamber, archegonial chamber, remnant of nucellus, female gametophyte and archegonia. So micropyle, it resembles like this. So here the sequence of structures from centripetal. Centripetal means from periphery to center. Fourth option 1. First is sclerotesta, then inner sarcotesta 4, then nucellus. Nucellus is present as a layer, and after the formation of seed it converts into perisperm. Then endosperm. Endosperm is nothing but female gametophyte present at the center. So outer sarcotesta, sclerotesta, inner sarcotesta, nucellus present as a thin layer, and endosperm. So answer is 1, 4, 2, 3. internal organization, character 1, character 2, you have to identify correct sequence. TS of leaflet, transfusion tissue, amphistomatous. Amphistomatous means equal number of stomata are present on upper epidermis and lower epidermis. So, TS of leaflets, transfusion tissue, amphistomatous condition is correct. TS of stem, siphonostele, TS of stem, siphonostele, broad medullary rays is correct. TS of choroid root, triarch condition, periderm present. TS of root, pentarch condition, root hair. Root hair are absent, periderm, pentarch is also absent, but TS of choroid root shows triarch and periderm. But answer is TS of leaflet, transfusion tissue, amphistomatous condition. Amphistomatous means equal number of stomata are present on upper epidermis and lower epidermis. Transfusion tissue is present, which is helping in lateral conduction. Just now we explained about transfusion tissue. TS of stem is siphonostele. Here the answer is 1 and 3, not first option, it is fourth option. TS of stem is not siphonostele. Broad medullary rays may be present, but it is not siphonostele. TS of choroid root, triarch is condition, periderm is correct is. So, answer not 4, it is 1 and 3. TS of root, pentarch, root hair is wrong. It is not pentarch, it is triarch and exarch. Archegonial chamber of cycas is formed by female gametophyte. Male gametes of cycas show their movement in archegonial chamber. If you draw the structure of well-developed ovule of cycas, it will be like this. It shows only one integument with a micropyle, a special sclerotesta at the center, a pollen chamber. This is 2R sarcotesta, this is sclerotesta. Another chamber called archegonial chamber, a remnant of nucellas like this, and a large female gametophyte, and 2 to 8 archegonia. So, this is the well developed ovule of cycas. So, the assertion is archegonial chamber of cycas has formed by female gametophyte. It is not formed by female gametophyte, it is formed by nucellas. It is formed by nucellas. Male gametes of cycas show their movement in archegonial chamber. Male gametes are formed in pollen chamber. Pollen tube is formed, siphonogamy takes place. Male gametes comes to the archegonial chamber. There they get released and swim here and there for some time. So, zooidogamy and siphonogamy both occurs. So, male gametes of cycas show their movements in archegonial chamber is correct. So, A is false. Why false? Archegonial chamber is formed from nucellas, not from female gametophyte. And male gametes of cycas definitely show zooidogamy in archegonial chamber. So, A is false, R is true. So, these are important questions related to Pteris, Funaria and cycas. All are important, some problems are discussed, important concepts are discussed in the form of questions, and this is about Funaria, Pteris and cycas. We discuss about next chapter microbiology, bacteria and virus in the next class, and then we discuss plant physiology.